digestive system. All questions answer solution. Enough for exam from this system. 1. Draw a well-labeled diagram of digestive system. Fig. Digestive system. 2. Name the organs of digestive system. The organs of digestive system are: main organs: mouth, pharynx, esophagus, stomach, small intestine, large intestine, anus. Accessory organs: salivary gland, liver, gallbladder, pancreas. 3. Name the structure forming different layers of GI tract. The structure forming different layers of GI tract are: mucosal layer, submucosal layer, muscularis layer, serosa layer. 4. Write down the functions of digestive system. The functions of digestive system are: ingestion is taking food into the mouth, eating. Secretion is the release, by cells within the walls of the GI tract and accessory organs, of water, acid, buffers, and enzymes into the lumen of the tract. Mixing and propulsion result from the alternating contraction and relaxation of the smooth muscles within the walls of the GI tract. Digestion: mechanical digestion and chemical digestion. Chemical digestion is a series of catabolic hydrolysis reactions that break down large carbohydrate, lipid, and protein food molecules into smaller molecules that are usable by body cells. Absorption is the passage of end products of digestion from the GL tract into blood or lymph for distribution to cells. Defecation is emptying of the rectum, eliminating indigestible substances from the GI tract. 5. Define peritoneum. Name the muscles of tongue. Peritoneum is the serous membrane lining the cavity of the abdomen and covering the abdominal organs. The muscles of tongue are: genoglossus, styloglossus, hyglossus, politoglossus. 6. Name the different parts of large intestine. The different parts of large intestine are: caecum, ascending colon, transverse colon, descending colon, sigmoid colon, rectum, anal canal. 7. Name the structures forming the wall of large intestine. The structure forming the wall of large intestine are: mucosa, submucosa, muscular layer, serosa. 8. Write down the functions of large intestine. The functions of large intestine are: absorption of some electrolytes and water. Feces formation. Feces consist of water, inorganic salts, sloughed off epithelial cells, bacteria, products of bacterial decomposition, and undigested parts of food. Microbial activity. Normally there are certain bacteria in our large intestine which form folic acid and vitamin B12. Elimination of feces. It helps in excretion of feces. Mass movement. Mass movement is due to gastrocolic reflex. When stomach fills, a strong peristaltic wave moves contents of transverse colon into rectum. The peristaltic wave is called gastrocolic reflex. 9. Define defecation. Write down the mechanism of defecation. The elimination of feces from the rectum is called defecation. The mechanism of defecation are: defecation is a reflex action aided by voluntary contractions of the diaphragm and abdominal muscles. The external anal sphincter can be voluntarily controlled, except in infants, to allow or postpone defecation. Gastrocolic reflex moves feces into rectum. Stretch receptors signal sacral spinal cord, which contract muscles of rectum and relax internal anal sphincter. External sphincter is voluntarily relaxed and defecation occurs. 10. Draw a well-labeled diagram of liver. Fig. Liver. 11. Name the different lobes of liver. The different lobes of liver are: right lobe, caudate lobe, quadrate lobe, left lobe. 12. Write down the functions of liver. The functions of liver are: liver regulates the nutrient metabolism, carbohydrate, lipids. Detoxifies the blood by removing or altering drugs and hormones. Removes the waste product bilirubin. Releases bile salts, help digestion by emulsification. Stores fat-soluble vitamins A, D, E, K, and vitamin B12. Stores iron and copper. Phagocytizes worn-out blood cells and bacteria. Activates vitamin D in its synthesis. Synthesis of plasma proteins. Removal of toxins. Synthesis and secretion of bile. Production of heat. 13. Write down the composition of bile. The composition of bile are: water and cholesterol. Bile salts I, E, Na and K salts of bilicid. Bile pigments bilirubin and biliverdin. 14. Describe the pathway of bile with figure. The pathway of bile with figure is described below. Bile capillaries carry bile formed in liver. From liver, hepatic ducts carry bile to gallbladder through cystic duct. From gallbladder, bile is carried to common bile duct through cystic duct. Common bile duct carries bile to hepatopancreatic duct, which drain bile into duodenum. 15. Describe the structure of liver. The liver is the largest gland in the body and the second largest organ in the body after the skin. The liver is divisible into left and right lobes, separated by the falciform ligament. Associated with the right lobe are the caudate and quadrate lobes. The gallbladder is a sac located in a depression on the posterior surface of the liver. 16. Name different types of taste buds with their specific taste sensation with figure. Taste buds have very sensitive microscopic hairs called microvilli. Different types of taste buds with specific taste sensation are: filiform papillae, sweet and salty. Fungiform papillae, salty. Foliate papillae, sour. Circumvallate papillae, bitter. 17. What is digestion? Digestion is the complex process of turning the food into nutrients, which the body uses for energy, growth and cell repair needed to survive. 18. Define peristalsis. Peristalsis is the involuntary constriction and relaxation of the muscles of the intestine or another canal, creating wave-like movements that push the contents of the canal forward. 19. Write down the mechanism of swallowing deglutition. The mechanism of swallowing deglutition occurs in 3 phases. They are: first stage, oral and buccal stage. The tongue moves upward and backward against the palate. Food is pushed into the pharynx by the tongue voluntarily. Soft palate closes off the nasopharynx. Mechanism of swallowing. Second stage, pharyngeal stage. Larynx rises so the epiglottis can close the opening of the trachea and bolus enters the esophagus. Third stage, esophageal stage. Upper sphincter of esophagus opens and bolus enters the esophagus, and a wave of peristalsis is initiated at the upper end and travels downwards, propelling the bolus downwards. As the bolus reaches the lower sphincter, it relaxes and food enters the stomach. 20. Draw a well-labeled diagram to show the pathway of the phases of gastric juice secretion. Fig. Pathway of the phases of gastric juice. 21. Give the composition of gastric juice. The composition of gastric juice is: water, mineral salts, mucus secreted by mucous neck cells in the glands, hydrochloric acid, an intrinsic factor secreted by parietal cells. Inactive enzyme precursors. Pepsinogens secreted by chief cells in the glands. 22. Describe physiology of digestion of food in stomach and intestine. The physiology of digestion of food in stomach and intestine is described below. Digestion in stomach. Food is mixed with gastric juice, which is secreted by the gastric gland. Food is converted into fine form by the muscular action of the stomach wall. Gastric juice contains HCl, pepsinogen and mucus. These are secreted by the peptic cells, parietal cells, and goblet cells respectively. Prorenin is found in infants. HCl converts pepsinogen and prorenin are converted into pepsin and renin respectively. It also kills microorganisms. Pepsin digests protein into peptones and proteases. Pepsin plus protein right pointing arrow peptones plus proteoses. Semi-digested food is in the semisolid form called chyme. It is passed from the pyloric stomach to the duodenum through the pyloric sphincter. Digestion in intestine. Chyme is mixed with bile juice and pancreatic juice. Bile from the liver is mixed with food. It emulsifies the fat, neutralizes the acidic food of the stomach and provides an alkaline medium for the action of pancreatic juice. Enzymes from pancreatic juice also digest different foods. Enterokinase from the intestinal wall converts trypsinogen of the pancreas into trypsin. Trypsin digest protein, peptones, proteoses into amino acids. Amylase digest carbohydrate into disaccharides. Lipase digest fat into fatty acid and glycerol. Trypsin plus protein plus proteoses plus peptones right pointing arrow amino acid. Amylase plus starch right pointing arrow maltose. Lipase plus emulsified fat right pointing arrow fatty acids plus glycerol. Intestinal juice is produced by crypts of Lieberkühn. It contains various enzymes like orepsin, maltase, lactase, sucrose, etc. which digest the remaining food. Peptones plus orepsin right pointing arrow amino acids. Maltose plus maltase right pointing arrow glucose plus glucose. Lactose plus lactose right pointing arrow glucose and galactose. Sucrose plus sucrase right pointing arrow glucose and fructose. 23. Write down the functions of pancreas. The functions of pancreas are: an exocrine function is that helps in digestion. An endocrine function is that regulates blood sugar. It produces hormones like insulin and glucagon. It produces enzymes like pancreatic juice. 24. Explain the mechanism of carbohydrate digestion, metabolism, in human. The mechanism of carbohydrate digestion in human is explained below. Digestion of carbohydrates starts from oral cavity. When ingested food is mixed with saliva in oral cavity, salivary amylase in saliva partially digests polysaccharide present in food to disaccharide. Then partially digested food passes to stomach. Gastric juice in stomach stop further digestion by salivary amylase I. There is no digestion of carbohydrate in stomach. Then the food reaches small intestine, where complete digestion of carbohydrate polysaccharide takes place. At first, remaining polysaccharide in food is digested to disaccharide by pancreatic amylase. Then the disaccharide is further digested to monosaccharide by specific enzymes, namely maltase, sucrase and lactase, as shown in chart above. Here the digestion of carbohydrate is completed. After complete digestion, monosaccharide so formed is absorbed into intestinal blood vessels through intestinal villi. Thus the digestion and absorption of carbohydrate takes place. 25. What is emulsification? What are the factors that help in emulsification of fats? When food reaches small intestine, fats in food is converted to digestible form by action of bile. The process is called emulsification. The factors that help in emulsification of fats are bile salts, lipase and phospholipids. 26. List the different endocrine cells found in stomach and mention their functions. The different endocrine cells found in stomach and their functions are A. Gastrin. Released by G cells in the antral portion of stomach. Actions. Stimulates HCl secretion and growth of stomach and pancreas. B. Secretin. Secreted by duodenal cells in response to increased acidity and increased fat. Actions. Stimulates secretion of bicarbonate by pancreas and biliary tract. C. Cholecytokinin, CCK. Secreted by duodenal eye cells. Actions. Stimulates pancreatic secretion of enzymes and gallbladder contraction and promotes growth of pancreatic cells. D. Glucose-dependent insulinotropic peptide, GIP. Released from duodenal K cells in response to luminal glucose and fat. Primary action is to promote insulin release. 27. Write down the nerve supply of tongue. The nerve supply of tongue are the hypoglossal nerves, the lingual branch of the mandibular nerves, the facial and glossopharyngeal nerves, the nerves of taste. 28. Draw a well-labeled diagram of tongue showing the dorsum of tongue. Fig. Dorsum of tongue. 29. Draw a well-labeled diagram of tooth. Fig. Tooth. 30. Write down the functions of tongue and teeth. The functions of teeth are incisors and canine teeth are the cutting teeth and are used for biting off pieces of food. The premolar and molar teeth, with broad, flat surfaces, are used for grinding or chewing food. The functions of tongue are tasting, gustatory sensation, chewing, aiding in mastication, speech formation, sound formation. 31. Write down the dental formula of primary and temporary teeth. 32. Write down the composition and functions of saliva. The composition of saliva are water, mineral salts, a digestive enzyme, salivary amylase, mucus, lysozyme, immunoglobulins, blood clotting factors. The functions of saliva are saliva lubricates food to assist deglutition, moistens the buccal mucosa, which is important for speech, provides an aqueous solvent necessary for taste, helps in sucking and suckling by a fluid seal, secretes digestive enzymes, e.g., salivary amylase, protection from microbes by saliva, i.e. antimicrobials. 33. Name different types of salivary glands. The different types of salivary glands are parotid glands, submandibular glands, sublingual glands. 34. Draw a well-labeled diagram of stomach.